Welcome to Biotech Academy. Today's topic is about active transport. Active transport molecules are transported away from the thermodynamic equilibrium. Hence, this active transport requires energy is required for the active transport. This energy, which is coming by the hydrolysis of ATP. This ATP, which is produced from electron movement or from light. The maintenance of the electrochemical gradient in biological system is very important to consume of almost all 30 to 40% of the total energy which is expenditure in cells. So total that cell is utilizes 30 to 40% of the energy through electrochemical gradient, the maintenance of the biological system by the electrochemical gradient. How it is ATP synthesized? This ATP is synthesized by the electron movement or from light. So electron movement, which electrons movement ATP is synthesized. This ATP is required for the active transport. Cells maintain a low intracellular Na plus concentration and a high intracellular K plus concentration. So cell outside the membrane outside we can see that Na plus concentration, that is low level of the Na plus concentration. Cell inside high level of the K plus concentration. It is along with the net negative electropotential gradient, that electron potential gradient by the ATPase activity. So ATPase activity, one ATP produces 3 Na plus and 2 K plus. ATP is an integral membrane protein which requires phospholipids for activity. So ATP works in the presence of phospholipids. The activity is required by the phospholipids. ATP is an integral membrane protein. Integral membrane protein. It is a membrane protein. Here the molecules are transferring, synthesizing through ATP. ATPase. This ATPase is an integral membrane protein. See, ATPase is catalyzed by the both ATP and Na plus and that cytoplasmic side of the membrane. This, see, we can see an example. This is membrane. This is outside of the membrane. This is inside of the membrane. See this, inside to outside, outside to inside. This is barrier by the ATP, ADP. Mg2 plus is required for the ATP function. ATP losses is one inorganic process and produces pulling of this potential gradient Na plus to the inside. Here 3 molecules of Na plus is going inside to outside. So 3 sodium ions are pumped to the inside and 2 potassium ions are pumping to the outside of the cell. So outside membrane potential by Na plus, inside membrane potential by the K plus. These is an integral membrane which is inhibited by the ovabine and digitalis. So these two or inhibit the ATPase activity, which is found in binding to the extracellular domain. So inhibition of the domain ATPase by the avabine and can be extracellular K plus ions. The blocking of the extracellular K plus ions. Na plus and K plus ATPase pump moves 3 Na plus ions from inside to the cell to outside and brings 2 K plus ions from the outside to the inside. Every molecule of ATP hydrolysis to produce ADP molecules. It is membrane associated ATPase. So sodium ions are pumping from inside and potassium ions are from outside of the cell. So each molecules which produces 1 ATP is produced. So 3 molecules of sodium ions and 2 molecules of potassium ions. So various molecules which are comparing from outside and inside of the mammalian cells. Na plus, K plus, A2 plus, Mg2 plus, chlorine, bicarbonate, glucose, proteins. These are different potential in extracellular fluids and intracellular fluids. The action of the Na plus and K plus ATPase activity is the primary source of the membrane potential for most of the eukaryotic cells. This we can say that electrogenic process. So most of the eukaryotic ATPases are P-type ATPases. Most of them ATPases are P-type ATPases, and some of them are carrying Ca2 plus. Many of them are carrying Mg2 plus. So ATP carrying Ca2 plus, which are transported to calciums inside of the cells to the outside of the cells, which return to the 2H plus from the outside of the ATP.